In this unit, we'll be looking at the pathophysiology of cancer or neoplastic disease, malignant neoplastic disease. We'll define that in a minute. Uh, Virtually all of us have been affected by this disease, either personally or somebody that is a relative or a friend. It'd be a rare person that would say that they have never known anybody to have had cancer. I picked these pictures just to show how terrible this disease can be. If we look at the woman on the right with her metastatic breast cancer and all that necrotic dead tissue that's growing, I mean that's obviously an advanced case. Looking at let's see if I can get my hmm, sorry about that. I was trying to get my laser pointer, but if we look at this down here, this is actually a piece of colon that was opened up. And this is this would be colon cancer. If you notice, it's gone through the whole wall of that tissue. Um, and this is actually an X-ray of lung cancer. And so this area that's circled here, that white area, the thing to remember is X-rays turn X-ray film, at least we used to use film, but now digital, X-rays turn things black. And so if we look over here in this area, that's pure black, pure black. There's nothing between the X-ray film and X-ray source. Where here we have white, which is bone and tissue, and so this area of density means that the X-rays aren't making it through, and there's a mass of something in that lung. Now you can't diagnose that it's lung cancer just by the X-ray, but you know that there's a mass of something in that tissue. So a quick preview of what we're going to cover. We'll look at terminology of cancer, uh, the types of cancers. We'll look at characteristics of the cells. We'll look at promoters and inhibitors. And then finally, we'll kind of summarize things looking at what's called oncogenesis, or the genesis or production of cancer, along with metastatic disease and how things spread, basically. And finally, we'll summarize with the causes of cancer and what do you see in cancer patients. This slide is getting a little old. The numbers aren't probably up to date, but that's not the reason I put this slide in. I don't want you to memorize the percentage of men and women that have certain types of cancers. But if we notice that there's different biology among cancer, and by that I mean some are more common and prevalent, where others are more lethal, if you would. And so if we look at prevalency of cancer, men, about 1 in 4 men, sorry, 1 in 4 men will get prostate cancer. But if we go down here, only 1 in 10 of those individuals will actually have mortality due to that, or death because of that. So in other words, prostate cancer is a very prevalent cancer. However, it's not the most lethal of cancers in men. And we look at lung cancer, that is pretty lethal. So we'll get 15% of men will have lung cancer, excuse me, 31% of cancer deaths will be because of lung cancer. So that one is much harder to treat because of the biology. And the same with women. Over here we'll have, the most common, again, about 1 in 4 of women 1 in 4 women will get breast cancer. Yet lung cancer again is the highest cause of death due to cancer. So breast cancer is number 2. So 15% of deaths due to cancer because of breast, yet 26% are because of are due to breast cancer. So if we look at the numbers, what that's telling us is that cancers are different. Some are much more aggressive and much more lethal than others, where others are maybe slower growing and longer lasting. For example, the prostate cancer in men. So let's start our journey in cancer pathophysiology looking at terminology. This can be very confusing, and oftentimes lay people don't understand things. They don't understand that there's different types of tumors. And so what we'll define these as we go along. So first of all, tumor is a very generic word. It all it means is a mass or swelling. And remember, when we went through the inflammatory response, we talked about tumor as being the swelling part of inflammation. And so again, that has no specific meaning as far as cancerous or not cancerous. And the same with neoplasia. Neoplasia is just new growth. So typically that's abnormal new growth. And it's going to, as we're going to learn, these cells are rapidly dividing and proliferating really unchecked, if you would. And so that would be neoplastic. And it literally means new growth. So neo is new and plasia is growth. But we have to define those. And so these neoplastic tumors are either benign, which are non-cancerous, or malignant, which are the cancerous ones. And they've got invasive properties. And I always say that normal cells make good neighbors. They grow, they may touch the cells next to them, or they do touch the cells next to them, but they stay home. They don't invade into that area, where malignant neoplasms are bad neighbors, and they want to take over the neighborhood, and so they actually will start invading territory and moving to distant parts of the body. Let's start by looking at the benign tumors, the non-cancerous tumors. So what do they look like? How do they behave? First of all, they contain cells that look like normal tissue. So if you were to take that tissue and take a slice of it, it would look like you would expect it to look. So for example, if it was a liver benign tumor, you'd slice into it. It looks like liver, or a cell at least that is similar to that. And they may actually perform the normal functions, and that's very common, for example, in endocrine tumors. The tumors that are benign tumors, they're normal tissue, yet they're secreting hormones excessively, and so we'll call that oversecretion. So again, we can have normal functions. So it can look normal and it can function normally. I mean, overfunction normally would probably be the way to put that. Most of them have capsules around them, which is really, really nice. So if you are a surgeon going in to remove this tumor, you can very easily see the edges of that, and so they're not as difficult to remove completely, depending on the tissue that's surrounding it, but they're encapsulated, and so I can remove that easily. They are good neighbors. Now they're growing, and they may cause some compression because of that unchecked growth that's going on, but they don't invade. They don't take over the neighborhood. They don't pilfer the neighbor's house and kick them out. So they're much more benign, right? Much friendlier than neoplastic tissue. I'm sorry, than malignant tissue. But like I say, they can grow fast enough that just because of that compression, that they can damage nearby organs, but they're not actually invading the organ. They're just compressing it. So let's look at an actual benign tumor. And so this is a uterine leiomyoma. We'll define that in a second. But needless to say, let me kind of give you a little background on what we're looking at. This over here is an ovary. The oviduct is going to run here. This would be the uterine muscle. There's the lumen of the uterus, and then again we have more uterine muscle. I believe that looks like an ovary as well in that area, along with an oviduct. So we're looking at really a section of the uterus, the full uterus. So this would have been a complete hysterectomy on this particular woman. And so we're going to define benign tumor. So this tissue is the benign tumor. So if we notice, it's got a capsule around it. It's demarcated. In fact, there's a second tumor that goes right here. Again, very demarcated. So we've got two tumors in this area that are benign. They're not. It's growing uncontrolled, but if we notice, it's not invading into, this tissue here. It's not invading into the tissue there, which we would see with a malignant tumor. The term uterine leiomyoma is some of the verbiage that I want you to be familiar with. And so what we're looking at here, is where it's from. So the term uterine, obviously this is a uterus, so it originates from the uterus. And then leiomyoma has a lot of meaning to it as well. So let's do the second half of the word first. Myoma. Okay, so myo is muscle, and oma is typically a benign tumor. And the term leiomyoma means it's a benign tumor of smooth muscle origin. And so a leiomyoma, a uterine leiomyoma, tells me a lot. It means that it's a tumor that originated in the uterus, and the tissue that was abnormal would be the smooth muscle of the uterus. So how are malignant tumors different than that? Well, first of all, they don't look anything like a normal adult cell. All the histopathology that histology that you learn in anatomy, the way that muscles look and the way that the intestine looks, the way that liver looks, these do not look like that. They are totally abnormal cells. And so if you don't know what you're looking at, you'd really have no idea where that tissue came from. The other difference is that they divide very rapidly, or more rapidly at least than normal tissue. And so tumors can grow quickly, at least quicker than normal tissue would. Again, we can have slow growing tumors, and we can have very fast growing tumors, aggressive tumors, but by and large they grow relatively quickly compared to other tissue. They also mutate faster, and they can actually change type. And this mutation, as we're going to learn in our antineoplastic unit, causes antineoplastic resistance. So it's very similar to antibiotic resistance, where the bacteria mutate and become have defenses against that antibiotic. Well, these cells can have the same thing, and so the treatment will work really well right away. And then we're selecting for these mutations that cause resistance, and so eventually the treatment isn't working as well, or maybe not at all. The other thing is these do not make good neighbors. The tumor does not have a clear boundary, so it's really, really difficult once the surgeon gets in there and they're trying to determine where the edges of these are so they can remove the abnormal tissue. And they've got these legs that kind of go out. In fact, the term cancer means crab, so it's like crab legs that are going out is what it's named for. These cells also do not perform normal functions, and they are pretty much abnormal altogether. Some of them actually will secrete hormones, which is associated with other tissues. The most common one, as we'll learn later on in this unit, is they commonly secrete a parathyroid-like hormone, which leads to a hypercalcemia. So the parathyroid hormone is naturally secreted by our parathyroid gland in response to low blood calcium, causes osteoclast activation and more calcium reabsorption from the kidney and the gut, and that brings up the blood calcium to the normal range. And so it's very common for cancer patients to have hypercalcemia, and they actually have a word for that called hypercalcemia of malignancy. So if you have a patient that comes in with some vague complaints and you run blood work and you find high calcium, right away you need to put cancer on your rule out list for that patient. And again, much like a benign tumor, they can grow fast and compress tissue, and more commonly they actually just invade and destroy the surrounding tissue. So again, very, very poor neighbors. So let's look at a cancerous uterine tumor. So we're going to call this one a uterine leiomyosarcoma. Okay, sarcoma. And so what we're looking at now, even I have a hard time seeing what we're looking at, but this tissue here is normal uterine tissue, believe it or not. That's normal muscle tissue, and this would be the cancerous tumor. Okay, surrounding it, and you really can't see a good edge. Again, we've got it here, right? Where there's not a, there's a big chunk of it there, but it invades into this area. So if you're perhaps it invades in here, and there's even going to be some legs that go off into this that are microscopic that you can't even see. So in cases like this, you do, again, a complete hysterectomy. You just take the whole thing out because it's really, really difficult to determine exactly where it is. And so again, we name the same way. Uterine, it came from the uterus. Lyo would be smooth muscle, and a myosarcoma. Now, a sarcoma means that it's a cancerous tumor. And so let's kind of move on, and we'll start defining what these things mean. So here's some really good rules for you to know on types of cancer. So when you hear somebody has, for example, cancer, and my mom will say this all the time, so and so was just diagnosed with cancer, and my first question is always, oh, what kind? Well, cancer. And I said, well, okay, what kind of cancer? And they say, well, breast cancer. Okay, well, so what type of breast cancer? Because there's multiple different types. So if we think of the tissue involved in an organ, most organs have their parenchymal tissue, the active tissue. So for example, let's look at a liver. They have their liver cells, okay? And so those liver cells can become cancers. But there's connective tissue that holds those liver cells together. And so those can also become cancerous. And then there's blood vessels through there. And so the blood vessels themselves maybe became cancerous. So the source could be the liver, but there's multiple, I should say the organ can be the liver, but there's multiple different cell types and tissue types that make up an organ. And so you can have cancer virtually of any of those different types. So this kind of are some general guidelines that will define what type of tissue came from that area. So the term, first of all, let's define a few things. And I'm going to draw underneath there. So the term oma, if it's just an oma, typically that is benign. Okay, so if it's just oma, that would be benign. If we put in front of it carcinoma, now that is going to become a cancer. Okay, and it's cancers of in this case, carcinomas would be cancer that originate with the epithelial tissue in that organ. So here we have another definition. Here, I'm going to erase that one so we don't get too many things going at one time. Whoops, a carcinoma, again, is going to be a cancer of epithelial origin. And adenocarcinoma, okay, so now we have two. So we know carcinoma, that is certainly going to be a cancer. And adeno is gland, right? That's what adeno stands for. So now we are going to have, again, epithelial tissue because we have that term carcinoma. But in front of it we have adeno, so we're going to call it glandular epithelial cells. So maybe it's a salivary gland or something to that effect that is made up of epithelial tissue. Next one would be sarcoma. Well, that's one on the previous slide. So sarcoma, okay. So if it's a sarcoma, it's a cancer, and that cancer is of what was mesenchymal tissue as an embryo and a fetus. Well, an embryo actually, because it's differentiated by the time you get to be a fetus. But mesenchymal tissue is undifferentiated connective tissue, and it differentiates during our embryological development into muscle and connective tissue, and therefore sarcomas are going to be cancers of the muscle and connective tissue. Now let's go into some other ones. Okay, so let me back up. And so that last one we had was a uterine leiomyosarcoma. So that tells me it is a uterus. Leiomyo is muscle, and sarcoma, because it's a muscle, we're going to call it a sarcoma. If it was epithelial tissue, that would have been a carcinoma. Now let's switch to our blood cancers, which originate really in the bone marrow. So these are really bone marrow cancers, if you would. So leukemia, the term leukemia is a broad term for cancers of white blood cells. Okay, so cancers of the blood, white blood cells. Myeloma are going to be more specific, and those are cancers of plasma white blood cells, so plasma cells. Then we have what we commonly call in human medicine lymphoma. Now this one kind of breaks the rules if we call it lymphoma, because oma, if we have the end of oma, that means it's benign. But this is the one exception that we use in human medicine. We call them lymphoma commonly, and that is a cancer of lymphatic tissue. Okay, so lymph nodes, spleen, those kind of things. The technical proper way for that to say would be lymphosarcoma, because remember, blood is connective tissue, right? And in fact, in veterinary medicine, we call lymphomas lymphosarcoma, but in human medicine, we call it lymphoma. So you just have to know that a lymphoma is a cancer, even though it doesn't have the proper one would be to call it lymphosarcoma. So what are the characteristics of neoplastic tissue? Actually, even both benign and malignant neoplastic tissue. So neoplasms proliferate and form new tissue. That's what they're named for, right? New tissue, literally new tissue, or new growth. They don't wait for signals from the body that new tissue is needed. So typically, and we'll go through some of the process of this, cells typically need to be told to divide. These are usually growth factors that do that. And these cells are going to ignore those factors, the signals to divide normally. They also ignore the signals to stop dividing. So we have ways for cells to proliferate and ways to stop cells from proliferating. And these cells ignore both of those typically, or I should say one or the other, or sometimes both. Oftentimes they don't mature or differentiate to the job of normal tissue. Particularly in malignant neoplasia, they definitely are not differentiated, and sometimes benign neoplastic tissue actually do a different job as well, or don't do it as well as they should. And then finally, they don't die off. Okay, so as we learned in our first unit of the year, cells typically divide so many times, they run out of telomeres and they undergo apoptosis and die. And these cells do not do that. In fact, we'll call that immortality. These cells become immortal cells, and so they don't die off. They continue to live basically as long as the host is living. So let's list some of the characteristics of malignant neoplastic cells. So these would be cancer cells. So as we said earlier, they're going to lack differentiation, and they don't contribute in a healthy way to body functioning whatsoever. If we look at these down here, we looked at this earlier, and this is a normal cell that we'd see in a pap smear, a normal squamous epithelium that is sloughing off of the cervix continuously as it's being replaced. Over here, now let's look at this word, a cervical, so that's where it came from. Squamous cell, that would be the source. And if you remember, it's an epithelial cell, so we're going to call that a carcinoma. So this tissue in the slide around here is that same tissue, okay, cervical squamous cells. But these and these do not look anything alike. They're quite different. And so if I was to just show you, if I was to show you this slide, if I was to look at this slide, I'd know right away what I was looking at. I wouldn't necessarily know that it came from the cervical region, but it would be definitely mucosal epithelial cells that I'm looking at. Where over here, if you told me to take a look at this slide, I would have no idea. In fact, we look at some of these. Here's a bizarre cell. Here's a single nucleus. There's another nucleus. So I have multiple nuclei in that particular cell. Look at this cell down here. It has 5 or 6 nuclei inside that cell. So these are definitely abnormal cells. It'd be very easy to say, yeah, these are not typical cells. These are cancerous cells. And again, cervical location, squamous cell origination, carcinoma is because they're epithelial cells. The other, I guess the second, sorry, I almost skipped that one. The second characteristics would be that they're unstable genetically. And I pointed out already that we have abnormal nuclei in these cells down below. They can have abnormal numbers of chromosomes, and we can have multiple nuclei, as I pointed out on that slide as well. The third characteristic is they don't die. They're immortal. They have the unlimited ability to divide. Normally, telomerase, telomerase is a gene that allows telomeres on chromosomes to be built. So in other words, normally when a cell it undergoes cell division, the telomeres are the binding site and they get clipped off. And so it's like the ends of your shoestrings with the caps on them. If every time you tied your shoe and you cut the end of your shoelace off, eventually you'd run out of cap and run out of shoelace, and you would no longer be able to use that shoelace. Well, these cells don't run out of shoelace. They continually make those telomeres, and again, can become immortal because of that. They also invade other tissues. So we define that, and we could say they lose their contact inhibition. So normally cells, when they touch other cells, they stop growing that direction, and so that's called contact inhibition. The second one is they lose their anchorage dependence. So they're not stuck to that one place anymore, and they can actually move from one organ in the body to a different place in the body to another organ, and that's defined as metastasis. So again, they lost their contact inhibition, they invade other tissues, and they can actually leave that organ and go somewhere else, which leads to actually number 5 because of that anchorage dependence, the loss of the anchorage dependence. They can migrate and have metastasis. This picture here, it's an interesting one, is to show, I wonder if I can, let me see if I can do this right. It's a hemangio, boy, this is going to take a long time, I'm sorry. Hemangio, I'm running out of room, so I'm going to say sarcoma. At least that's what I was worried that it was. And so looking over here, this is actually a splenic tumor. So if you notice, that does not look like a spleen whatsoever. Down here looks like normal spleen tissue, but it's about the size of a volleyball almost. And this actually came from a golden retriever, so a big breed dog, but not that big, right? That's a pretty big mass. And hemangiosarcoma, the reason I put this slide in there, and I want to talk about hemangiosarcoma, is that particular tumor in these animals. By the time you see them, they've metastasized. So virtually there's 100% metastasis. Well, 99%, I should say. It's never 100%. And they go to specific areas. You'll always see it. They go to the right atrium of the heart. They metastasize to the liver and the lungs. And so those are 3 spots that they commonly go to. And so the treatment for this was to remove that liver. So we removed the liver. Remove the spleen. And while you're in there, you look at the liver. Well, this liver happened to be clean, so it wasn't a problem. We sent it off to the diagnostic lab, the pathology lab, and they looked at it. And it was really kind of nice. I wonder if I can get, I can get rid of that. So this dog ended up having a hemangioma, which was a benign tumor, which is, if we notice, it was very well demarcated right there. You can see where that demarcation is. And so this was a benign tumor. And a hemangioma, a hemangiosarcoma, is actually a tumor of the blood vessels in the spleen. And that's that hemangio term. And hemangiosarcoma would be the cancerous form, and this happened to be a hemangioma, which was the non-cancerous form. And the final characteristic that we'll look at is that they can divide without growth factors. They ignore chemical signals. And so we have really, and we'll talk about 2 different types of growth factors. One is the stimulatory growth factors that tell cells to divide, and the other are inhibitory growth factors that tell cells to stop dividing. And so in this slide over here, we're looking at mitotic figures in this tissue. And so we're looking at abnormal, here's the one that's undergoing mitosis there, dividing, but there's another one right there, there's another one there, there's another one right there, another one over here, another one up here. I mean, there's just all sorts of mitotic figures in that tissue. And so that has got a very, well, they call that a mitotic index. So that tissue has a high mitotic index. In other words, there's lots of mitosis going on compared to the number of cells in that particular view. And that is an indication, again, of dividing regardless of growth factors. So let's take a look, dig a little deeper in these growth factors. They're known as mitogens, right, because they cause mitosis. Normally, the number of cells produced equal the number of cells that die. So in other words, the total number of cells in a person's body remains relatively constant once you've matured, obviously, and reach adulthood. So to understand this, let's kind of look at the cell cycle. We'll review this. Hopefully this will be a quick review for you. So over here we've got the G0 phase. Okay. So notice we're going to have the G0, G1. We've got this entrance right at this point. We have the S phase, the G2 phase, and then we undergo mitosis, or the M phase. So G0 is the cell at rest. That cell is not dividing. It is doing its job in the body. So, for example, a liver cell doing what liver cells do, metabolizing things, secreting things, whatever it is that they tend to do. Then when they're told to do this, so growth factors will come in and give this cell a signal and say, it's time for you to divide. We need more of you. And so it's going to enter into our G1 phase, basically, as we're looking at this. So we'll enter through and go through the S phase. Okay, so the S, the G stands for gap. I should probably tell you that G stands for gap. Then we're going to enter the S phase. Normally I like to get you guys participating in this and ask, well, what goes on during the S phase? Well, S stands for synthesis. So in the S phase, we are actually dividing our DNA. We're making another set of DNA so we can divide those between two cells. Then we're going to undergo this checkpoint here to check that DNA out. That's going to be on our gap number 2. And if everything looks good during this interphase thing, so we made the DNA, we've checked it. Now we're going to enter the mitotic cell division phase, and we're going to double our cell. And then we can either go back to the G0, or we can continue through that cycle, making more and more and more cells, depending on what the signals are and what the body needs. So let me kind of go into a little history of these growth factors before we get into the mechanisms of them. Growth factors were discovered by these two individuals in the upper right-hand corner who won the Nobel Prize in Physiology and Medicine in 1986. So Dr. Stanley Cohen and Dr. Rita Levi Montalcini. Dr. Cohen was doing research in wound healing, basically, as I understand it, and looking at what stimulates that epithelial production. And it was known at that time that mouse saliva actually allowed wounds to heal a little bit quicker. So there's some factor in that he discovered. And it was a heat-stable protein, and he isolated it from the mouse saliva, and he named it epidermal growth factor. And so that was a seminal event in the history of learning about cell division and actually cancer, but he didn't know that at the time. Dr. Levi Montalcini was actually looking at, was a neuroscientist, and she was looking at what stimulates nerves to undergo mitosis in the cell cycle during embryogenesis and brain development and nerve development. And so she discovered a thing, and she called it nerve growth factor. So both of these have become extremely important. Well, these just the category of growth factors are extremely important in neoplastic events because they're going to stimulate proliferation, differentiation, and affect survival of cells. So how do they work? Well, these are proteins, again, proteins, so they can't make it through the cell wall. And so they are going to bind to a receptor, and and this receptor is specifically a tyrosine kinase receptor. So if you look down here, here's the receptor right there. So the growth factor will bind to the receptor, and it's going to stimulate inside the cell, so it's going to transduce that signal, and it's going to activate what's called tyrosine kinase. They actually, if you notice on the right-hand side, drop this off, we can look over here. They actually dimerize, and this is the actual way that they function. There should be two of them that connect. But be that as it may, they are going to cause a cascade of events to occur in the cell, which stimulates our cell division, our entrance into the cell cycle. These are all the growth factors up here. They're circled. You don't need to know all of those, but there's many, many different growth factors that we're aware of. We're actually discovering more and more as we go along. But if we notice up here, we say EDF. All right, that's epidermal growth factor. That's the one that Dr. Cohen discovered. And so because of that, if we notice, there are 4 different types of receptors. In fact, maybe my computer's kind of slowing down here. Well, maybe I won't do that. It's not allowing me to get in. Okay, sorry for that pause. So if we notice down here, there's 4 different types of receptors, and they're numbered. The first one we're going to call epidermal growth factor receptor, so EGFR. But really, it's also known as, so I write this out, HER1. Okay, so HER1 stands for human epidermal growth receptor 1. And so we also have a HER2, human epidermal growth receptor 2, HER3, HER4. The reason I want you to know that is targeted therapy. These HER receptors are a target for that. And so, again, this is an area of treatment that we can use. So that's how they work. They activate these things. They trigger cell proliferation. They allow blood vessels to grow in the tissue. They're going to inhibit apoptosis, so the cells live longer. And when we've got a mutation of this sort, it allows migration, adhesion, and invasion. Well, that's cancer right at that point. So again, these are the very, very, very important factors that we have called growth factors. So cancer is truly a genetic disease. So we have mutations or abnormalities of cells which originate in the genes. And so what is the basis for these abnormalities? So we've got really 2 different categories of genes that are important in oncogenesis or cancer production. The first we're going to call proto-oncogenes. So these are oncogenes that by the way, these are normal genes. Okay, so proto-oncogenes are normal genes, and then we have our tumor suppressor gene is the second category. Again, a normal gene. So these genes are used in cell production and division to either stimulate or suppress entering that cell cycle. So in proto-oncogenes, again, these are normal genes that code for things that are important. So the growth factors we just talked about, the growth factor receptors that we just covered, the signaling pathways that are going to go on inside the cell, again, enzymes that produce secondary messengers, nuclear transcription factors. So they're very, very complex. There's all sorts of cells, all sorts of enzymes, all sorts of proteins that are required in a cell to allow that entering that cell division cell cycle. So these are going to promote the cell cycle. So proto-oncogenes cause the cell cycle to go, where tumor suppressor genes do the opposite. They're going to inhibit the cell cycle, promote apoptosis, and decelerate that whole process. So what are some of the abnormalities that can occur? The first one over here, let's see if we can get here, okay. First one here is abnormalities in growth factors. And so what's happening now is, I've got these growth factor receptors. So these HER receptors are up here. And normally another cell is going to secrete that and tell it to continue growing. Where in this case, we've got a mutation in the chromosome here that's going to cause its own secretion of hormone, growth factor, and that growth factor is going to come over and stimulate the growth factor receptors. So we're going to call that an autocrine signal. Okay, so production of growth factors, production of mitogens, remember that's another word for them. So they're going to have an autocrine type pattern. Another thing that can happen is we'll have an abnormality in a chromosome that activates a gene, and the proliferation of that gene, the gene starts making more receptors. Okay, so now we have abnormal growth factor receptors. And so these black growth factors are not necessarily the growth factors that are normal for that cell. You know, these red ones up here should be activating the cell, but since we have an abnormal receptor, those things are stimulating cell proliferation. So we can, and there's several different things like that. We can mutate and the factor. Mutate the receptor itself. Or we can change the affinity of that to external, or we can increase the numbers of those. So there's all sorts of abnormalities, genetic abnormalities that can cause problems with the receptors, these HER receptors or growth factor receptors. If we have abnormal intracellular pathway components, in other words, these receptors aren't active at all. There's no growth promoter, no mitogen that are activating those tyrosine kinase receptors. But down here, we've got this cascade that's occurring anyway. Okay? And so this is the cascade. This is really what these receptors normally would transduce into that area, the signals. There's that cascade of events, again, stimulating that cell to produce more and more cells. So those are activating mitosis, basically the cell cycle. The final one we have down here would be abnormal transcription factors. So transcription factors are things that activate transcription down here. So in other words, we're going to transcribe the DNA, we're going to make messenger RNA, it's going to start making proteins. Okay, so here's the proteins that are made from this abnormal transcription. And basically this activating that transcription factor is going to spur the cell to enter the S phase, the synthesis phase. And so now we'll be duplicating our DNA at a time when we really shouldn't be. So if proto-oncogenes are normal and tumor suppressor genes are normal, what happens when they mutate and those abnormalities that we just discussed occur? We're going to change the name of that, and now we're going to call them oncogenes. Okay, so onco is cancer, genes are obviously the genetic code. And so they're still going to code for the proteins needed for cell division, but they're going to do it abnormally, just as we just discussed those four different type of abnormalities that we could see. So the effect of the oncogene is we can promote too much of this protein. It could have an abnormal protein, so it could activate things when it really shouldn't be. It could be a protein that turns on all by itself. A protein that is being made even though it shouldn't be, it's not being signaled to be made, right? So those tyrosine kinase receptors aren't activated, but the protein's activating the cell cascades. The proteins that can't turn this off, so that now we have just constantly repetition of cell division. And we also can make some proteins that should be made by a different cell, again signaling our cell to do something that it shouldn't be doing. Well, how do these mutations occur? So what can cause this normal proto-oncogene to become an oncogene? And there's four overarching categories of etiologies in this. Number one is viruses, or are viruses, I should say. And so the retrovirus is a very common one. Retroviruses can actually insert their genes. So we have a retrovirus For example, like the human papilloma virus, the Rous sarcoma virus would be another one. There's a few in veterinary medicine, the feline and bovine leukemia viruses. There's a simian retrovirus in monkeys. So there's several documented viruses, retroviruses that are actually oncogenic. And so these retroviruses get into the cell. They're RNA viruses. They cause reverse transcriptase to make viral DNA, and then that viral DNA gets inserted into our chromosomes, activating gene expression that shouldn't be occurring, making proteins that shouldn't be made. Uh, the second main one would be some kind of a mutagenic event. Okay, so at this point we're gonna and we'll talk about mutagens in a little while. So it could be a chemical, it could be radiation, right? Something that causes that mutation. And what's going to happen at that point is we are going to mutate the proto-oncogene and activate it and turn that into just a simple oncogene, right? So that's a direct mutation at that point. A third way to make it is to mutate a regulatory sequence. So again, we have this mutagen coming in, a chemical or radiation. And so this green area is the regulatory gene that either turns on or keeps off this oncogene, this proto-oncogene. And so if we turn this and activate this proto-oncogene, the proto-oncogene actually isn't mutated. It is the regulatory sequence that controls that proto-oncogene that's mutated and basically has the same effect. It turns it into an oncogene because now it's expressing things continually that it shouldn't be. And then the final step is we get proto-oncogene amplification. So we get multiple mutations of the same thing and amplifying that effect. So a single mutation is not that big a deal. I mean, it can be, but it doesn't necessarily have to be. But if we get protogene amplification, now we have lots and lots of product being formed, causing cell abnormalities. Now let's take a look at these tumor suppressor proteins, right? So tumor suppressor genes that produce these tumor suppressor proteins. This is a little more recent for us. We gave the Nobel Prize for growth factors in 1986. We gave the Nobel Prize in physiology and medicine in 2001 for the discovery of these. And so these are the researchers in 2001 that received the Nobel Prize. So we have Hartwell, Hunt, and Nurse, basically. So Leland Hartland, Timothy Hunt, and Paul Nurse, who won that. Hartwell, these were all separate researchers looking at the cell cycle and discovering these things. Hartwell discovered this. The first one we're looking at is called a checkpoint. Hunt discovered what's called the cyclin-dependent kinases. So that'd be the second one we're going to talk about here, the cyclin inhibitors. And Nurse actually discovered cyclin itself. So let's look at these three discoveries and see how do they come into play in cancer. So the first one is the checkpoint. The checkpoint usually stops division in mutated cells. So if there's something wrong with this cell, the cell is going to stop what's going on and either fix it or cause the cell to undergo apoptosis and stop the progress altogether. So these are going to keep most of the mutations that we have. I mean, we have cells mutating every day in our bodies, but they don't develop into cancer, and this is one of the ways that we can protect ourselves from that. The second one is that these proteins that control the checkpoints, I'm sorry, not the proteins that control these checkpoints are called tumor suppressor proteins. So these are the genes that are expressed in making those proteins to do that. And they're called cyclin inhibitors. And we'll cover that in a second, what cyclin does and how it works. There's several parts of this in the cell cycle. The classic example is the P53 protein, which is expressed from the P53 gene. That one actually has a name called the guardian angel gene. So we'll discuss that in a little bit. But if we mutate these, okay, so if we mutate these suppressor genes, now we're going to call those oncogenes as well. Okay, so these are mutated normal genes that don't function, don't stop the cell division at these checkpoints like it actually should. So the checkpoints that are controlled by these tumor suppressor genes, well, actually there's two of them, and one is there's a checkpoint between our G1 phase and the S phase. We'll call that the G1S checkpoint. And there's another checkpoint between the G2 and mitosis phase, so we'll call that the G2M checkpoint. I think I may have we may not have gone through all the steps of what goes on in the cell cycle in that previous slide. I don't remember now. So let me just repeat that. If I have, I apologize. But G0 is the resting phase, so the cell's doing its thing. I remember talking about that. The G0 phase, when activated by those growth factors that we discussed, it'll enter into the G1 phase. And the G1 phase is where we'll see cell growth, and we'll start producing enzymes that are required for the synthesis of DNA. So really, where G1 is, we're preparing for a replication. Between the G1 and the S, we have this checkpoint where the cell will pause, and it's under regulation by this thing called cyclin E. Okay, so cyclin is going to regulate all of these. And so cyclin well, we'll talk about cyclin in a second. Let's suffice it to say that what happens here is we're going to look at the DNA before we replicate it to make sure that there's no mutations that we are going to replicate and carry on. Then if everything is fine there, we pass the G1 checkpoint. Then we enter the S phase, which is the synthesis phase. So now we are replicating our DNA. So we're making our excess chromosomes that we can split into two. So we're going to make it make the DNA for both cells. And then once we made that, we enter the G2 phase, and then the G2 phase, we are synthesizing the microtubules that are going to be used to pull those chromosomes apart, right? So we replicated them, and now we have to somehow separate them into two different cells. And so now that we've replicated it, made the microtubules, before we undergo mitosis, our prophase, metaphase, anaphase, telophase, in other words, we split into two cells, we have another checkpoint to make sure that everything's normal, that the DNA replication is okay, the microtubules are okay, everything is what we needed. And so that's the cell cycle. Those are the two checkpoints that we have. Now, cyclin is what controls this, and cyclin is a family of proteins that are going to control what we call cyclin-dependent protein kinases, CDKs. So again, cyclin-dependent protein kinases, and then the CDKs are what control the cell cycle entry. Okay, and so these are the ones. So for example, our P53, our guardian angel gene is one of these CDK proteins that we're dealing with. And so the ones that control this, the cyclin, the predominant cyclins in each of these cell cycle. So in G1 entering into S, G1 cyclin E is the important one. Once we get to the S phase and G2 phase, cyclin A is responsible for all of that activity that goes on. And then during our entry through the checkpoint into mitosis, cyclin B is a prominent family of cyclins that control that. So what happens there is, again, they regulate this. They make sure that everything's right. They've checked the DNA to make sure that that's duplicated properly. And then we also see whether or not the cell is large enough, right? We have to get it big enough so we can split it into two. So that's really what cyclin regulates in the cell cycle. All right, so let's summarize what happens. Okay, and so in the cell on the left, so this guy right here, this would be a normal cell, and this cell right here would be an abnormal cell. So what we're going to do is have a mutation, or we're going to lose our P53 proteins. So I taught this many years and didn't really look at it close enough, but I noticed that there's actually facial expressions on these cells. So we have a nice happy cell here. Everything's normal. And over here we've got kind of a sad face cell that just things are mutated and not quite normal. So in a normal cell, as we activate this, if we damage the DNA, our P53 gene is going to bind to the DNA and check that. And so um, as we enter our cell cycle, I should say, we've got a couple of things that can happen. Uh, One is that we're going to kind of stall the cell cycle at that point, allowing DNA repair. Okay, so that's this area here. So we're going to stall the cell cycle, see if we can come back, fix the cell, and if that's the case, then we're fine. We get a successful repair. We're normal, so now we can undergo our mitotic change, or we can actually again arrest it in G1 and repair that as well, right? And so now we come back and we'll fix the DNA in G1 normal, and we'll undergo mitosis. If we can't repair the DNA, we fail, then what's going to happen is we're going to go down this pathway, and we're going to activate our apoptotic genes, and we're going to actually undergo apoptosis or cell suicide at that point. So that would be a cell, this cell up here, which has what we're going to call proto-oncogenes, normal proto-oncogenes. Now let's look at this guy over here, okay, which has an oncogene. As his DNA is damaged, he undergoes the same, or he or she, I should say, it undergoes the same pathway, but now we can't activate our P53. So no cell cycle is arrested. We can't repair the DNA. Nothing happens, and now we have the mutant cell that actually undergoes mitosis. Okay, so now we have an abnormal cell, and now do we not only have a sad face as the progenitor, but now we've got this evil face down here, this cell. So that's actually a malignant neoplastic cell that's going on at that point. So again, no repair was done, and so now we have oncogenes because it replicates abnormal cancerous cells. So I'd encourage you at this point, I can't show this, it's a video, but you're in YouTube now, so just open another YouTube window and search for how tumor suppressor genes block cell division McGraw-Hill, and watch this video. It's a very nice video on the mechanism by which that these things work. Another mutation that we'll see is a mutation that we referred to earlier about the telomeres. And telomeres are where the polymerase enzymes attach to the DNA, and once they attach, they destroy those base pairs in the DNA and the ends of the chromosomes, basically. And so if the end of the telomere does not get duplicated, well, let me rephrase that. It binds there, and that end does not get duplicated, and therefore the chromosomes continue to shorten and shorten and shorten as the cell divides from parent cell to daughter cell. And so if it doesn't do that, right, let me rephrase that. So the question is, what would happen if it doesn't do that? So the question on the bottom is, a man has a mutation that causes some of his cells to rebuild their telomeres after every division. Is this a good thing or a bad thing? Well, the answer would be that's a bad thing, because now the cell will continue to divide forever and ever and ever. And so here's a picture down here on the right-hand side, kind of showing what the ends of the chromosome looks like, and it lands or binds there, but doesn't replicate that portion of the cell. So let's put it all together. Those are some of the mechanisms, or at least some of the principles of those mechanisms. And so oncogenesis is actually the production of cancer, the genesis, creation of cancer. And so we have three phases that occur with this. Number one is initiation. Second is promotion. And then finally, third is progression. So it's a multi-step process. And so in initiation, typically a mutation activates a proto-oncogene or inhibits a tumor suppressor gene. So in other words, we create an oncogene, and then we get proliferation of that cell over and over and over. And this mutation occurs using carcinogens, and we'll go through a few of those a little bit later. I think we all know about them. You know, light infrared radiation is one. So the sunlight that's coming down on us. X-rays would be another one. Radiation in general is another one. There's chemicals that do this. So for example, a lot of industrial chemicals, just the carcinogens in general. Smoking obviously is one that's a very common carcinogen. A lot of different chemicals that actually initiate mutagenesis in the cells. So then after initiation, we get to promotion, and promotion is really proliferation, where now the mutated cells. So here's our first initiation. There's our first mutation, and now we're promoting or we're growing in these cells at this point. So we typically at proliferation we have activation of other oncogenes. So in other words, we inactivate tumor suppressor genes or activate proto-oncogenes, causing that. We'll also see some factors that go with it. For example, nutritional factors, infections that are going on at the same time. Those can actually be promoting events as well. So we promote growth to that. And then finally we get to the progression where now we start exhibiting malignant behavior. So now we're starting to eat our way down through the basement membrane. So here's those crab legs, if you would, that are growing out. We've invaded the surrounding tissue around there. We've actually gone into the lymphatic system or blood system, and now we're metastasizing to different areas. And then we stop in distant areas, and we actually start a new tumor at that point. So this would be the progression phase, and it surrounds and metastasizes at that point. So this ability for these cancer cells to leave and go to spread throughout the body, which we call metastasis. There's kind of some special things go on at this point. So up here we have cells that are kind of in place at this point. Here would be a normal cell abutting its partner cell next to it, or its neighboring cell, I should probably say. And they're connected. These cells are connected by cadherins. So that's the connective tissue that's holding these cells together. And then here's the basement membrane. So this would be an epithelial cell that we're looking at in this particular case. And then down here, or say in the middle, the picture we have here, notice that we've got a cell that has dissolved the cadherin. So there's enzymes that will go ahead and dissolve that cadherin. So we're going to break it down. And so now we've got this rift. The cell is kind of changing at this point. And we're going to start digesting the collagen. So normally the collagen fibers sit down here making up the basement membrane, and this type 4 collagenase is being released, and that's going to start digesting the collagen basement membrane, allowing it to generate a hole, if you would, down the bottom. And then in the next picture down here, we have these receptors on the bottom of the cell. And so receptors are going to pull the tissue through using laminin and fibronectin receptors. So they're going to kind of grab and start migrating its way through down I could probably draw here, right? So it's going to start migrating down. Well, we'll try that again. There we go. It's going to start migrating down this direction. Okay. And then in the final picture down here, we've made it all the way through basically. So we've gotten into the tissue below the basement membrane, and now we can enter the blood or lymphatic system and migrate to take seed and take hold in distant tissues. Some cancers are actually hereditary. So in other words, they're not a mutation in the individual, they were actually a mutation in one of their ancestors. And so because of this, it's hereditary or heritable. and so it can be spread from parents to children. A couple examples of these. One is very, very famous. It's the BRCA gene. And the BRCA gene is actually there's a BRCA1 and a BRCA2. BRCA stands for breast cancer. And these are tumor suppressor genes that are mutated and passed on, and there's a percentage of the population that carries those. It'll give you a very high risk of having breast cancer at a relatively early age, and so many of you have probably heard of families the women in many families who have died very young of breast cancer. And so there's actually an example that Francis Collins talks about in his book, The Language of God. And for those that don't know who Francis Collins is, he was the Human Genome Project Director at the University of Michigan. He's actually a physical chemist, a quantum mechanics, but before well, I guess as he finished his PhD, he decided to go to medical school and became a medical geneticist. He is currently the director of the National Institutes of Health, so he's done quite well in his scientific endeavors. At any rate, he's a Christian. Wrote a book called the Language of God, and in that he tells a story about early on one of the first patients that he helped with his medical genetics was a woman that was going to have a prophylactic double mastectomy because her siblings and mother had all died at a young age of breast cancer, so she assumed that she was going to do so as well. So Dr. Collins went ahead and convinced her to do a genotype, and they ran that and found out that she did not have the BRCA gene at all, and so she was no more at risk of having breast cancer than anybody in the public, right? So the population as a whole, and so she was able to she was prevented from having to have that prophylactic double mastectomy. So there's some real winning situations that we can have if we know some of this information. Another one is the retinoblastoma gene, and again, that's a heritable gene. It's a tumor suppressor gene, just like the BRCA gene is a mutated tumor suppressor gene, and the RB gene is actually, we know it very well. It's on chromosome 13, right at this point. So if we notice, we've got one normal allele. Okay, so that one's not mutated, and we have one mutated allele. Which means that patient is safe because it's actually a recessive trait. If once both your parents are carriers of this mutated gene and you become homozygous mutation like this one over here, now you're at risk. So this one here is homozygous for the RB gene, which puts that individual in a great risk of having a retinoblastoma cancer or tumor at any rate. Then there's the RET gene, which is different than the other two. That's the first two are tumor suppressor mutations. This one is actually a mutated proto-oncogene that's associated with thyroid carcinoma, medullary thyroid carcinoma. That is actually of the calcitonin secreting cells. Calcitonin is the hormone that lowers blood calcium in the body, trying to tone it down, if you would. And RET stands for rearranged during transfection. That's where they got that term. But at any rate, it's a proto-oncogene that mutated into an oncogene, and it's actually a receptor. The tyrosine kinase receptor gene is what it is in that case. So it's a mutated tyrosine kinase receptor. So, are there things in our environment that can cause cancer? Well, absolutely, there are, and radiation is one of the biggest ones. So, for those of us that like to go out in the sunshine and feel its warmth and tan and all those kind of good things, especially we think of on spring break, for example, after a long winter. So that is actually very carcinogenic, particularly those tanning booths that we tend to like to go to. I as well as others like to call them cancer booths, right? Because they're very prone to skin cancer. And the problem is we do this when we're young typically. And so we expose ourselves to way too much ultraviolet light, either in tanning booths or in outside, and we burn our skin, and that sets up that initiation phase. And so if you remember, it may take many, many, many years before we see anything. So typically, the things that we do when we're in our teenage years, our 20s, our 30s, set us up for these skin cancers when we get into our 60s, 70s, 80s. And so it's, even though it may not occur to you when you're young, it certainly, you may regret it when you become much older and start fighting some of these cancers. The picture on the other one is X-rays. X-rays can cause mutations, and so here, this picture over here is actually the very first X-ray. This is a copy of the X-ray that Wilhelm Röntgen, who discovered the X-ray basically, and he took this in 1885, a little before Christmas in 1885, and this is actually this is an X-ray of his wife's hand. Now we've come a long way in X-raying. I mean, it's very poor. You can tell that there was a lot of X-rays going on through this, because look at how black that area is. And so this is actually it looks like a negative now that I'm looking at it. But at any rate, this was the first one. It's really kind of hard to see the joints, and you can't see the metacarpals here very well at all yet, so we refined it a whole lot. But the point behind this is that there is no safe level of X-rays. They are all mutagenic. Every time you get an X-ray, you could cause initiation and promotion of cancer. The good news is our digital X-rays today is much, much, much less powerful than the X-rays that we used to be exposed to. There are chemicals that can cause cancer in people, and so there are really two that we categorize as either direct reacting agents or indirect reacting agents, or procarcinogens, or initiators of cancer. So the classic example of those are polycyclic hydrocarbons. That's what's found in tobacco smoke. So that's what we find in cigarette smokers who are at risk for cancer. The soots and the tars that are in there as well, and the oil. In fact, it's if you barbecue grill and get charred, charred meats. I remember my mother used to love grilled chicken, but she wanted the skin charred and blackened. And that actually, I took a drug metabolism course from a professor who her area of research was actually in looking at what that charred food, the effects in the body, and looked at the chemicals and realized that actually can be a procarcinogen. Almost wrecked grilling for me, but I overcame it, and I still grill. I just make sure I try not to char it very much when I do that. They're industrial agents, so things that we'll see in the workplace. So things like benzene, asbestos. Asbestos is no longer used, at least in pipes and things, but it used to be in houses and public buildings. And again, that causes particularly lung cancer. Although it's in the news again now, because the talcum powder, some people are saying talcum powder contains asbestos, and therefore has been causing ovarian cancer in women. That is being argued, and we don't know whether that's actually the case or not. But we do know asbestos will cause ovarian cancer. That we do know. There's some insecticides, fungicides, so pesticides can cause that as well. So farmers and exterminators, those type of people can be exposed to that. So they need to wear protection so they don't initiate cancer in them. And food and drugs. So we talked about grilled food. Well, smoked foods are the same way. There's an aflatoxin, which is a fungal toxin. So if you get foods that have fungus in it that's producing aflatoxin, that can be carcinogenic. And then we'll talk about anti-cancer drugs in our next unit, and many of these anti-cancer drugs are actually procarcinogens as well. And so it's becoming much more common that we put people through a regimen of chemotherapy, and then we cure that cancer, but then 10 or 15, 20 years down the road, Another one comes back, and it was initiated by that initial chemotherapy regimen that we put the patient on. We talked about viruses, so these are some of the viruses I'd like you to know about that can cause cancer. So hepatitis B and C, which we'll cover in the liver unit, they may cause liver cancers. We have the Epstein-Barr virus, which may cause Burkitt's lymphoma, so that's our mononucleosis virus. Our HIV virus, the AIDS virus, causes Kaposi's sarcoma, which is a skin cancer that is really only seen in patients that are severely immunocompromised, like HIV patients. And then the human papillomavirus, which we've shown several pictures, or at least a couple of pictures in this class, that can cause cervical cancer. And down here is a picture of a cervix, and that big nodule there, that whole area is actually cancerous, and most likely caused by the human papillomavirus. So let's take a look at how that occurs in the human papilloma virus when they infect this cell. And so if we look we're in the G2 part of cell growth. Okay, so here's our G1 initiation. We're making our enzymes. We pass the checkpoint. We synthesize our DNA, and now we're growing and producing our microtubules. And we get before the G2 checkpoint. This is what we find in this particular case. And so we have, if HPV is not present, okay, so this is not, so this would be the normal situation. As we enter our cycle, the P53 gene, right up here, P53 protein, will find that mutation that has been caused by the retrovirus. And I'm sorry, which would have been caused by if there's a mutation. And it won't allow those cells to divide, so that occurs at that G2 checkpoint, and you get a healthy cervix at that point. If you are infected with, or at least the epithelial cells of the cervix are infected with HPV, then we end up inhibiting the P53. So P53 is inhibited by the genes that HPV inserted. Therefore, the abnormal cell that was replicated with abnormal DNA can multiply. And now we've got two abnormal cells. They'll multiply with mitosis to the 2. Then they'll go to 4. They'll go to 8 to 16, and now you've got uncontrolled growth into the tumor. So again, that is basically what occurs. So the G2 checkpoint fails in the cervical squamous cell carcinoma, which is actually another good one. Remember we talked about cervix, that's where it's located. The squamous cell is the cell that is abnormal, and carcinoma means it's of epithelial origin. So we're getting really to the end of our cancer unit. We've looked at what the mechanisms of cancer, the genetics of cancer, some of the mediators of the cell cycle. So what do you see in the patient? So what occurs in these patients that have them? That by them I has cancer. Well, first of all, you'll have changes in organ function. Okay, so we're going to damage the organ. We'll have an inflammatory response that goes on with that because we've got dead and dying tissue, and we'll end up at the very end. The organs will fail because we'll basically replace the organ, or at least a significant portion of the organ, with abnormal cells. We can also have immune suppression. So now we're overburdening the immune system. The immune system's battling the cancer, but it can't work. Or perhaps it's a tumor that causes is an immune tumor. So therefore, it's the immune system itself that's being suppressed. We'll have local effects. So again, as we can compress nerves or veins, GI obstruction if it's a large tumor in the bowel. And so local effects would be things like bleeding, compression of blood vessels, compression of lymph vessels. You'll see edema, ascites, an effusion that's going on because of that's happening. You'll get compression of hollow organs, compression of nerves, as we said in that. So you can see pain, paralysis, those types of things that will go on with local effects. Then we'll see ectopic hormones secreted. And in fact, we talked a little earlier about the parathyroid-like hormone that can be secreted, increase so we'll see that hypercalcemia of malignancy. Uh, but they have other things as well. They may provide, well, we call this paraneoplastic syndrome, and so hormone-like proteins are being released, and some of the proteins that we see that mimic these hormones. One would be antidiuretic hormones, so we'll see water issues. Adrenocorticotropic hormone, so we'll actually see some adrenal medullary type of effects that are going on in these patients. The parathyroid-like hormones, we talked about the malignancy, I'm sorry, the hypercalcemia of malignancy, and some cancer cells will actually produce proteins that affect clotting, and so it's we see a lot of different things because of these secretions that the tumors are producing, causing these paraneoplastic syndromes. And then finally, as the cancer progresses, you get these really protein wasting and breaking down. And here's a picture of an unfortunate individual they're in a negative energy balance. The tumor is taking all of their energy. Thus we call it cancer cachexia. Cachexia is starving, basically. They may not have an appetite even. So not only is their metabolic rate high, but they're not hungry. They don't feel like eating at all. And it also affects the bones. The bone health disappears, so we see fractures and those types of things secondary to this cancer cachexia. Well, it's kind of a depressing way to finish a lecture talking about cancer cachexia, so I thought I'd bring about a little bit more, something a little lighter, looking at the African plain and a family of elephants. Again, as always, we'll have a question review, so if you have any questions, this is one of the more complex units, lots of moving parts, and to be honest with you, there's lots of things that we don't understand. And by we, I'm talking about the medical and scientific community. Right now, there's lots of research going on in this area. So bring any questions, and we'll see you in our next meeting.